Hi, my name is Roni Cohen-Fultheim from the Lebanon lab at Bar-Ilan University, and I'm here to ask you, how does our genome avoid self-activation of innate immunity? My poster deals with this question in different ways. Our genome contains inverted repeats that, when transcribed, can form double-strand RNAs and trigger cellular innate immunity. These double-strand RNAs can exit the nucleus into the cytosol, where they can be detected by double-strand RNA sensors. A-to-I RNA editing can be detected by sequencing. A-to-I RNA editing prevents recognition by cellular double-strand RNAs. Adenosine deaminates into inosine, which denatures the double-strand RNA and prevents the recognition. In my poster, I show a proof of concept in collaboration with Professor Yuval Dor from Hebrew U in type 1 diabetes. Type 1 diabetes is an autoimmune condition in which the immune system attacks the beta cells in the pancreas. What happens is that beta cells get distracted and there's loss of glucose control for the patient. In this experiment, our collaborators created an ADAR1 knockdown in pancreatic beta cells and showed that it mimics early type 1 diabetes phenotype. Our proof of concept shows that the knockout of the active exon in ADAR1 causes a linear decrease in the exon expression, looking at wild-type, heterozygous, and knockout genotypes. In parallel with that, in correlation with that, we can see that the editing decreases also linearly, which is very nice and not always common, between heterozygous knockout and wild-type. After that, we can see that the phenotype achieved is a loss of glucose tolerance in the knockout mice. And in addition, there's insulitis of the beta cells and the pancreas, and it increases with time. So basically, we have ADAR knockout, which brings us to early type 1 diabetes phenotype. In my research, I look for allo elements that can cause this reaction. What we do is we look over all allo elements, over 1,100,000 elements in the genome across the GTEx portal, the Gene Tissue Expression portal, and we look for all editing levels. For each allo element, we count the adenosines and the guanosines for all adenosine positions, and obviously for all other mismatches as controls, and we pool all these to account for one value per element. So that's mismatch counting. After that, we pool the counts according to different groups. All GTEx donors are pooled into one tissue, and all tissues are pooled into one donor. In addition, we pool all the database into one single pool, which basically means we can look for every single element. We have four values, one representing sample per sample, which is tissue and donor, one value representing the editing per donor, one value representing the editing per tissue, and one value representing the editing in the whole database, tissues and donors combined. Now I want to show you, first of all, a few of our results. The first thing we can see is that we can filter these allo elements to make sure we have a good signal-to-noise ratio. What we filtered by was we chose to look at the maximal noise, which is not A to G, or for other mismatches, the relevant mismatch, or the complementary. For A to G, that means T to C, or for example, for C to T, that would be G to A. And we ignore the complementary mismatch, but we look for the other maximal mismatch possible. We demand that this mismatch would be at least times X less than our editing. Here you can see that if we do that, then we decrease the number of elements slowly, which basically means that the more we demand for the noise to be lower than the editing, the less elements we get. That's on one side. The other side we want to look at is the specificity. We want it to be specific sensitivity. We want it to be sensitive for editing. So what we ask for is we ask that the element will contain at least 5 sites or any other number. What you can see in the graph is different parameters of that. 5 sites that are at least 1% edited, where the editing is contributed by 2 mismatches from 2 different donors. At the very least, it can be more. So what you can see in this plot is on the x-axis, you see combinations of all these parameters. What happens when we increase the demand for noise to signal ratio? What happens when we increase or decrease the demand for sites? What happens if we demand 2 sites? What happens if we demand 5 sites? That's on one hand. The other thing we demanded was for no other mismatch to be very dominant. Up to 3 sites where A to G editing index, where their index is, where the non-A to G editing index is up to 30%. So you can see here how how these parameters act, and you can see that the more we increase the base, aside for a few different, a few changes at the beginning of passing on to the next noise, to the next site threshold, you can see that mostly it is a linear reduction. The more we increase and make the parameters more strict, the less elements we get. However, for the non-A to G elements, for the next best mismatch, this is very constant, which basically means our specificity and sensitivity are very high. Next thing we do is we take these elements, and you can see here a subsample of the distribution in different tissues. You can see different tissues like muscle skeletal, which are very low edited. You can see the distribution is very more towards the left, while you can see other things like artery tibial and artery aorta that are very spread out, which basically mean they have many elements that have high editing. 2 examples of highly edited ALU elements. One is this ZNF, zinc finger, that has, you can see the IGV here. You can see in the IGV picture here lots of editing. All the blue is basically C to T, T to C. Okay, it's the C's that you can see. It's basically editing on the minus strand. And what you can see here is the double-strand RNA structure. Look how nice and long it is. And you can see also the UCSC genome browser. You can see the 2 elements. In blue, you can see our element, and in Y, you can see the inverted ones. And you can see they're very, very close. Another element, which shows a much higher editing, but this is this is intergenic. You can see the A to G editing index, where you can see G's on the IGV marked as orange, and on the bottom you can see the original A's. You can see the double-strand RNA structure is much more, right here, you can see it's much more tight and long. And you also see the UCSC picture, where you see there's 1 element we found and 2 elements that are inverted. So this is my poster. I thank you, and if you have any comments, I'd like to hear. Thank you for listening.